Okay, so my name is Ferriol Calvet, and I'm here to present the Universal Protein Coding Gene Finder. We did this work at the CRG and the UPF in Barcelona, and we are working under the Catalan Initiative for the Earth Biogenome Project. To start with this presentation, I would like to introduce you to gene annotation, which is the process in which we obtain the coordinates of genes present in a given genome and assign a function to them. This process is key for the usability of genomes. If we have the genome annotation for a given gene, we're able to do posterior analysis. Otherwise, the analyses we can do are very limited. When it's time to generate a gene annotation, there are different approaches, and among them, we highlighted two of them, gene annotation pipelines and ab initio gene predictors. Inside gene annotation pipelines, we find those of Ensemble, NCBI, or other big institutes, and also each individual group can create their own customized gene annotation pipeline. And in the field of ab initio gene predictors, we can see also like many different programs. Among them, there are Breaker2, Augustus, Geneid, GeneMark, and many others. And now we'll see how they compare. So gene annotation pipelines produce annotation of a higher quality as they use multiple sources of information and also a lot more resources in terms of analysis, which comes to the drawbacks where they are expensive in terms of time and resources because they usually need experimental data, which is costly and needs to be generated and analyzed. And still it is incomplete because we know that we will never be able to generate a full complete gene annotation of a given genome. And then if we move to ab initio gene predictors, the good thing of these programs is that they are fast. They have very low requirements as they usually only require a parameter file and the genome of the species that we want to annotate. And they are less complex in the sense that they are easier to run in general. And the other good point that we selected is that they capture protein coding genes. However, if we move to the drawbacks, we see that they can only usually annotate a single transcript per gene. They cannot annotate any of the non-coding genes, and they require parameter training. And also, they generate an annotation of a lower quality. So our goal in this project was to tackle some of these disadvantages here, and we started by the first one. So we wanted to see whether the fact of predicting a single transcript per gene made a big difference in the process of assigning functions to those genes, and when also like focusing on protein coding genes. So what we did is take 20 species in Ensemble and compare the functions assigned to a given gene when using all of its transcripts and when using only the longest transcript. And we call these two groups Ensemble and longest CDS. And here would be an example of a real gene. And we did this comparison by comparing the Go terms assignment of each of these situations. And what we observed is that both Ensemble and longest CDS are almost equivalent for the number of genes with Go, for the mean number of Go terms assigned to each gene, and also in terms of similarity. So with this first analysis, what we concluded is that the fact of predicting a single transcript per gene is not a drawback anymore. Of course, it's not a positive thing, but it's not a big problem. And we can, with this, we forwarded with the different topics. And in order to address them, what we did was produce a new gene annotation, ab initio gene annotation gene predictor, which we call Geneid. So this Geneid is made of the combination of Geneid for its gene prediction capabilities and Diamond for protein to DNA alignments. And the combination of these two programs allows us to produce a fast, open source, and easy to use program. This is how this program works. So it receives a genome sequence and a set of proteins, which can be provided by the user or automatically downloaded by the pipeline. And the first thing that it does is it runs the alignment within the proteins that were provided as input and the genome to annotate. And with this, we generate a file where we have these matches that will be used as hints for the gene prediction process down here in number 3. Then the second step that we do here is, in order to overcome the step of parameter training, what we do is we estimate which are the coding and the intronic regions of these supposed genes by using these protein matches of the same protein. These protein to DNA matches of the same protein. And with this, we generate the parameter file. So there are some matrices in this file that can be generated by using these estimations in here. And once we generated the parameter file and we have the hints, we can use the genome sequence, Geneid, and these two files generated here to obtain the final protein coding gene annotation. This is how it works inside. And from outside, what we can see is that it is very easy to run. So the first thing that we would need to do is to install Nextflow and Docker or other container software. It can be done with these two commands, just like this one here, and then downloading Docker from this website. And the next thing that you have to do is just type this command. So run genidx with Docker and then provide a genome and the tax ID of the species that you want to annotate. So until here, this is how our program works. But what we needed to do is show how our program works in comparison to others. So we know that ab initio gene prediction programs are not perfectly accurate, but we want to provide an ab initio gene predictor that is at least comparable to the best methods out there. And what we did is compare these 6 different gene prediction programs in terms of structural annotation accuracy, functional annotation accuracy, and execution time. And we did it for 105 vertebrate species annotated in ensemble. We use these species because we used ensemble annotation as a reference to measure how good or how bad were our predictions. In terms of structural annotation accuracy, we computed 2 metrics, sensitivity and specificity, both at nucleotide and exon level. Just for simplifying all this view, we will focus on the average value here. And what we see is that in terms of nucleotide level, Geneid that you can see here in green is slightly better than Augustus in red. But when we move to exon level, we can see that Augustus is predicting the exons slightly better than Geneid. So in this first part, we cannot say that Augustus or Geneid is better than the other, but we can see that they are among the top 2 and they are better than all the others. Then moving to the functional annotation accuracy, what we can see here is that Geneid is better in specificity and sensitivity as defined by our analysis. It is not really as straightforward as before to define sensitivity and specificity, but we used, again, ensemble as reference, and we tried to define these 2 metrics. But then when we move to the similarity between the Go terms assigned, so this is all about Go terms. And when we move to a similarity between the Go terms assigned to a given gene in Augustus and the same gene in ensemble, then we compute the similarity, and we do the same for Geneid. So we assign Go terms to a gene predicted by Geneid, and then we measure the similarity between those Go terms assigned to that gene and the Go terms assigned to that same gene in ensemble. And what we see there is that Augustus is better than Geneid. So again, the same thing happens. We cannot clearly define whether Geneid or Augustus is better than the other in terms of functional accuracy. But when we move to the execution time, what we clearly see here is that Geneid is almost two orders of magnitudes faster than Augustus. And this really is a big difference between these 2 programs, making Geneid much more efficient than that. Here are some examples of where we use Geneid. And this is a portal that we're producing with all the annotations of all the species. And these are some projects in which we contributed. Then the final take-home message is that for initial annotation efforts, we should prefer efficient computational methods. Geneid can be run very easy in 2 steps, as I showed before. And our tool empowers genome researchers all around the world to annotate their species with almost no requirements. So with this, I would like to thank all the people involved and thank you for your attention.